Hello, this is Brett Gidner, and I'm going to be going over today's slides on the evidence that gene regulation is a big part of how life works on Earth. So the key genetic basis that tells us that gene regulation has to occur is an idea called genome equivalency. This literally means that all cells in an organism have the same genome in them. Now, how do we know this to be true? A century ago, scientists thought that different cells in the body acted differently because they had been given different parts of the genetic material during development. Red blood cells got the genes needed for blood function, liver cells got genes needed for liver function, and so on. That turned out to be a wrong notion, and this slide shows some of the key evidence. The paper that we went over in class, along with today's notes, provides the evidence, and the key researcher on that paper, John Gurdon, many years later was a co-winner of the 2012 Nobel Prize in Physiology or Medicine for the work in part that was reported in this paper. Stem cells and regenerative medicine are direct offshoots of these early experiments. So as we discussed in class, what they did here was to transfer nuclei from different cells of a frog embryo or potentially a mature frog into an unfertilized frog egg to see if that nucleus could in fact allow for full development. And they found that some nuclei worked and some did not. The work of Gurdon and others predated a much more famous cloned animal, one called Dolly the sheep. Why do you think she got so much more press than Gurdon's frogs? Well, Dolly was in fact the first cloned mammal, and we are mammals. Dolly was cloned using the nucleus from a mammary gland tissue cell. Please note that the term cloning and cloned, as we're using them today, do not refer here to cutting and pasting together DNA sequences. Rather, they are referring to cellular cloning, the transplantation of the genetic contents of one cell into another. The fact that not all the nuclei from different tissues in a tadpole or an adult frog worked when put back into a nucleated egg is not because genome equivalency is not true. Rather, it reflects the fact that eukaryotes package their DNA in such a way that it influences gene expression. This is not the case in bacteria. Sometimes the packaging of eukaryotic genes can get in the way of using a gene right after you make the nucleus, when you transfer a nucleus from one cell to another. Why and how this occurs is a very hot topic these days. Here is the abstract of a recent paper that gets at this topic of DNA packaging and how it can influence events for many cell generations and even organismal generations after the event. Similar papers have been published showing that this is happening in humans as well. This has some significant implications for medicine and many other areas, including the philosophical concepts of free will and fate. What if something that happens to me today is not because of something I did, but because of something my parents did, or even my grandparents? Not everything that happens in an individual's body is due to the actions of that individual, or even to events during that individual's lifetime. That's a lot to wrap our heads around, and we'll come back to that later in this section of the course. Now, the evidence for genome equivalency goes beyond animals. Here's another classic experiment in support of genome equivalency, this time from the plant kingdom, where you can take a carrot root and sterilize on the surface sterilize it to get rid of bacteria and fungi. You can take out a slice of the root, mash it up, grow the cells in tissue culture, and then if you treat them with some specific hormones, plant hormones in this case, you can actually induce cells to start to form somatic embryos, very much like what would happen inside of a seed. And from those somatic embryos, you can get back a young seedling and eventually grow back in a complete carrot plant. So the bottom line here is most of the time, all the cells in the body of an organism have the same genome as the zygote that gave rise to the body of the organism. Now, there are some exceptions. In a few animals, in this case shown on the top left is a chordate called a hagfish, and on the right is a particular fly in the group called the sciarid flies. You see some chromosomes lose all the chromosome, or large parts of those chromosomes, but only in somatic cells of the body, meaning the germline still maintains the entire genome, but certain somatic tissues have lost some of the genes. And notice at the bottom of the page are some references in case this is of interest to you. In a few other animals, parts of chromosomes, but not entire chromosomes, are lost in body cells during development. This event actually happens in two cells of our own, our B cells, which make antibodies, and T cells, which make T cell receptors. Now, both these cells are unique in that they rearrange some of their DNA as they develop, and they do that in a way that's unique to each particular B cell or each particular T cell, meaning no two B cells act the same. And these DNA rearrangements lock those B cells and T cells into one particular type of gene expression that allows them to express either a unique antibody or a unique T cell receptor. Very interesting outcomes. So, if most of our cells have exactly the same genome, how do we account for different cell types and tissues acting differently? Well, the answer is an idea called differential gene expression, meaning that any given gene has its own unique expression pattern. This slide shows some data in support of differential gene expression. Total RNA was isolated from different frog developmental time points and from different frog tissues. That RNA was spotted several times within a column of what's called a dot blot on a special piece of paper. Then horizontal strips were cut out and exposed to a DNA probe for a given gene of interest that we're just calling genes A through F here. What we see is that most genes are expressed during a particular developmental period and/or in a particular tissue. There are only a few genes that are expressed all the time. Now, we need to always keep in mind that differential gene expression is not the total answer, as is mentioned here in this quote from three of the big names in the modern day development of understanding how bodies of animals and plants develop. But that said, genetics and genomic tools that we now have at our disposal allow us to look at the expression of one or many genes, and this is an incredibly powerful way to understand development. Our story about gene expression begins in all organisms, E. coli, and how it changes its expression to switch its use of sugars as carbon and energy sources. And that's where we will begin in the next class. Thank you.